Topic Notes 3.4, Measuring Primary Productivity. This is a picture of a macroalgae called Caulerpa. The genus name is Caulerpa. And it's also commonly called feather algae. Now, oftentimes somebody will see this in the sand flats and the seagrass beds in our local area and think it's a plant, but it is actually an algae. It's not vascular. And it's an example of a primary producer. Now, we are going to be talking about measuring primary productivity today. And in the ocean, it can be a little tricky because Caulerpa here, not so difficult, but most primary productivity in the ocean occurs with phytoplankton. So let's get into it. Our big ideas here: scientists can apply a variety of techniques to gather productivity data and investigate energy flow through an ecosystem. Now, not all biomass is available for consumers. Now, this is because some is oxidized during respiration to provide energy, and thus that's lost as heat, right? And then some is stored, adding biomass. Now, remember when we talk about phytoplankton, we don't have a lot of stored biomass because they have got a high turnover rate and they're very small. But something like seagrass is a little bit more traditional, where you do have a decent amount of biomass stored up in new blades as they're formed. So these are just things to keep in mind as we move forward. Now here's some terms. Gross primary productivity is the total gain in energy or biomass per unit area per unit time by autotrophs. So we're talking about producers here, right? So that's the total energy gained. Now, net primary productivity is the gain in energy or biomass per unit area per unit time by autotrophs after, here's the difference, after the deduction due to respiration. Now, in this diagram I've set up here for you, you've got phytoplankton. Obviously, they're primary producers, right? They are going through photosynthesis, they're creating glucose, they're adding biomass, but some of that is taken away through respiration and it's lost to the system. And whatever's left over is available to the next trophic level, which in this case would be other zooplankton like copepods. Now, we are talking primary productivity. There is something called secondary productivity where the copepods obviously are going through respiration, they're losing energy, and whatever's left over is available to the next trophic level, which happen to be little silversides that might pick them off in plankton, and then so on and so forth. So just keep that in mind. These concepts often stretch beyond just dealing with primary producers. Now we can put this in an equation, and here it is. NPP, net primary productivity, equals gross primary productivity minus R, which is respiration. Now in the graphic below, it kind of goes through what this process looks like. Now we of course have our phytoplankton right there in the green box to the left. And if you notice there, it's getting inputs from a few things. First of all, it's getting a supply of nutrients from upwelling. It's also getting a supply of nutrients through general recycling, meaning that other animals that die pass on, decomposition, things like that. So all this nutrient uptake is going into the phytoplankton, and of course then it's getting sunlight and carbon dioxide and stuff, and it's going through photosynthesis and it's creating biomass, right? But then it's using biomass for autotrophic respiration, and that's that green arrow within the green phytoplankton box. So if you notice, gross primary productivity is all of these things combined. Now, as you go out of the green box to the right, you'll see net primary productivity, which is essentially taking away autotrophic respiration. And whatever is left over is your net primary productivity that is available to the zooplankton and the bacteria in the little browner box towards the right. Now, in zooplankton, the bacteria, all that stuff, they are going to go through processes of living and dying and all that kind of stuff, and they're going to add nutrients back into the system that can then be used by phytoplankton later on. So this gives you just another visual to understand the system that we're talking about. Now, an ecosystem's net primary productivity is the rate at which plants accumulate dry mass or actual plant material. Now, in the ocean, it is expressed in grams of carbon per square meter per year. Now, net primary productivity is then available to be passed on to the consumer. Now on the right, you'll see a map that shows net primary productivity globally. And you'll notice that there are certain areas that are essentially productivity deserts where there's like nothing going on, which actually is a lot of the ocean. And then you'll notice around coastal regions and polar regions, you have more productivity going on. And we're going to get into why that is as we go, but it's nice to make that note right here. Now here is an example problem that you could theoretically get. Net primary productivity off the coast of Peru is 300 grams of carbon per meter square per year. Approximately 120 grams of carbon per meter square per year is lost through respiration. What is the gross primary productivity? So here you go. You have MPP equals GPP minus R. We want to find GPP. So we start plugging in the data as you see below. 300 equals GPP minus 120. So we rearrange the equation, we divide, and we get 420 grams of carbon per meter square per year of gross primary productivity. Now that we understand productivity a little better, let's talk about measuring it. Now, one way is through rate of photosynthesis. This can be determined by examining the change in either oxygen or carbon dioxide concentrations. When photosynthesis is taking place, carbon dioxide concentrations will decrease and oxygen concentrations will increase. Essentially, during the daytime, this is what's going on, right? Because photosynthesis is taking out carbon dioxide and it's producing oxygen as a byproduct. You can see that in the graph to the right. Now, at night, things change, right? Carbon dioxide levels go up, oxygen levels go down because it's mostly just respiration going on. No oxygen production is there. Now let's talk about an example about how to actually calculate this. And this is called the light and dark bottle method. Here you have two bottles, both filled with water that has phytoplankton in it. The one bottle is clear and allows light to enter it. Essentially, a bottle where photosynthesis can occur. The dark bottle, no light can penetrate and thus no photosynthesis can occur within the bottle. Now, if we measure the amount of oxygen in the bottles at the start of the experiment, at the end of the experiment, you're going to get different values. In the light bottle, we can start off with 8 milligrams of oxygen. In the dark bottle, we start off with 8 milligrams of oxygen. At the end of the experiment, after a certain amount of time, let's say an hour or two, the light bottle has 19 milligrams of oxygen and the dark bottle has 2 milligrams of oxygen. This shows us a few things. In the dark bottle, no photosynthesis is going on, so oxygen is not being created, it's being used up through respiration. In the light bottle, you definitely have photosynthesis going on, and thus your oxygen levels keep increasing. Now with this data, we can now calculate net primary productivity, respiration, and gross primary productivity. Net primary productivity would be essentially taking the light bottle end results, which would be 19 milligrams of oxygen, subtract it from the initial starting point, which is 8 milligrams, and you get 11 milligrams. That's what's available to the next trophic level, so to speak. Respiration, you would take what you started with, 8 milligrams, and subtract what you ended with in the dark bottle, which is 2 milligrams. So we lost 6 milligrams. In gross primary productivity, you would subtract 19 milligrams, which is what we ended up with in the light bottle, from 2 milligrams, which is what we ended up in the dark bottle. And that leaves 17 milligrams per decimeter cubed per hour. So some applications to this. Now, phytoplankton can be kept in a bottle, but it's a closed system, which is not actually what happens in the wild, right? Now, when the bottle is in light, both photosynthesis and respiration take place. When the bottle is in the dark, there will only be respiration. By measuring the dissolved oxygen levels in both light and dark bottles, you can calculate that rate of photosynthesis. Now, this technique can be applied to investigate the effect of light intensity on productivity. What you do is you take your light and dark bottles, you collect them, of course, and you place those light and dark bottles at the same depths they were collected at. and then you measure the dissolved oxygen as things go along. Now, this allows us to figure out that essentially productivity increases near the surface water with the maximum concentration of chlorophyll. And then productivity decreases as the amount of light limits photosynthesis the deeper you go. Now, the compensation point is the light intensity at which the rate of photosynthesis and the rate of respiration are equal. Now, on the right of this graph, you'll notice the sunlight available as it decreases with depth, which makes total sense. Now, productivity follows that same line. It's higher at the surface. As you go deeper, that red line skews down and becomes less. You'll notice respiration stays relatively steady throughout, but there's the point where the red productivity line crosses the respiration line. That's the compensation depth. Below that, respiration exceeds what's being produced in terms of oxygen. Now, at the compensation point, there's really no change in the amount of carbon dioxide or oxygen. The net productivity would be 0 because of that. Now, below this depth, producers can't survive because the rate of respiration would be greater than photosynthesis. 90% of marine life is found above the compensation depth in what we call the euphotic zone, where there is light. So another method of measuring productivity would be looking at changes in biomass. Now, measuring the rate of accumulation of biomass per unit area can give us quite a lot of information. In order to do this, you have to harvest the producers after a set amount of time and generally dry them out. The reason why we do this is because water can be variable in living things from one point in time to the next. So if you take that water out, it makes things a little bit more consistent in terms of comparing masses. Now, we can do this in quite a few different ways. This is a little bit better for shallow water, seagrass, macroalgae, that kind of communities, because you could go out there with quadrats and transects and you can measure this stuff. However, we also, when you're dealing with phytoplankton, this is a relatively tricky method to apply. Now, I included the picture to the right of the fisheries because we often talk about biomass in terms of primary producers, but we also tend to talk about biomass in terms of secondary biomass when it comes to other trophic levels. And we do look at fisheries data to try and gauge at least an inkling of an idea of how much biomass might be out there. Now, it's a very imprecise science because we don't know what the collection methodologies are, things like that, but we try and get some information and we can tell if fisheries are decreasing or increasing based on catches, annual catches, things like that. But I just wanted to throw that in there as an aside. We are focused on primary production right now, but secondary production is out there as well. Now, there are some limitations to using biomass as a measure of productivity. First, it only measures net primary productivity as the plants continue to go through respiration. And also, it doesn't account for the biomass consumed by herbivores, like this pesky green sea turtle munching on seagrass, taking our data away. Now, another increasingly important method is using satellites that can indicate chlorophyll content in relatively shallow depths. It allows for large-scale monitoring, which is really great to really have a global perspective. What we find is most productive areas are found in tropics and at the higher latitudes. The least productive areas tend to be where there is smaller supply of nutrients from deeper waters. Here in this image, you can see chlorophyll concentrations globally. And again, look at how there's really concentrated in the polar regions, and there's a few areas elsewhere, especially near in coastal regions, things like that. Now, there are some limits to this. There's the relationship between chlorophyll and the concentration in the biomass is not fixed, so it depends on the individual species, so that can change. And also, satellites can only indicate relatively shallow depths. So it doesn't account for the entire eutrophic zone. So again, but it does give us a good image to monitor relatively efficiently. Now, I know there's a lot of supporting details with this one, and so this main idea may seem simplistic, but we kind of went on for a while, I know. But remember, at the end of the day, it's all about this. Scientists can apply a variety of techniques to gather productivity data and investigate energy flow through an ecosystem. Until next time, keep learning.